Hello, I'm Pat Byrne, a faculty member in Soil and Crop Sciences at Colorado State University. My area of research is plant breeding and genetics, and I'm especially interested in the use of wild crop relatives for improving our crop plants. I'm standing in an experimental wheat field at our research farm known as Ardec, north of Fort Collins, and I would like to talk to you about the evolutionary history of wheat and how that affects how we might use wild species, wild collections, to improve wheat. So to my right here, we have one of the ancestors of wheat. This is Triticum urartu. It is known as having the A genome. It's a diploid. It has one set of 7 chromosomes. And you can tell it's kind of rangy. These seed heads are going to fall over. And they have a trait known as shattering, where the seed heads will fall apart into many distinct seeds. So not a very useful trait for a crop. We like the seeds to stay together. So that's the A genome, one of the ancestors. This is another one. This is Aegilops speltoides, which is close to another of the ancestors. It has the B genome. You can tell this is very prolific. It has lots of seed heads. They're kind of skinny, but a lot of seeds produced on that plant. So what happened in evolutionary history is a cross between this A and the B genome plants to produce a hybrid that had the AB genome constitution. That AB plant was selected and adapted over time to produce durum wheat. And that's what's shown here between them. Durum wheat is where we get pasta. And these plants are very big and robust. The seed head is very large and productive. And this is a crop that in the US is mostly grown in North Dakota and Montana and into Canada. Now, another hybridization took place in forming bread wheat. The AB genome, as represented by durum wheat, crossed with another species known as Aegilops tauschii, the D genome. And it was the combination of those three that produced our productive bread wheats that we know now. And here's an example of one of them. This is a very productive and popular cultivar in Colorado known as Byrd, B-Y-R-D. And you can tell there's just a lot of grains produced here, and it's a variety that is very adapted to the different kinds of conditions we get in Colorado. So let me show you at least one example of the D genome. Right here, this is one of a large collection of D genome plants. And I will try to explain something about the diversity that we have out here. The least diverse of the genomes within modern wheat is considered to be the D genome. So there's been a lot of research attention at looking at how can we increase that diversity. So what we've done out here is to plant 42 different types of Aegilops tauschii, the D genome, collected from a wide range of habitats, everywhere from Syria to China, and many countries in between. What we're looking for is particular traits that could be beneficial when crossed into bread wheat. Now, most of these look pretty weedy, and they are.
They are not currently in any form to be a cultivar, but the challenge here is to separate the potentially useful alleles or genes from everything else in the genetic background.